is that cell reproduction and understanding cell reproduction is critical for understanding cancer. Now, the question that my lab started is how, given this is such an important process, how is it controlled and how can those controls be investigated? Now, the next slide will surprise you, because the way in which my lab approached this was to take a very simple organism, yeast. You're used to yeast making bread, making wine, making beer, but we also use it to experiment and find out about the nature of life. And in this case, this is a fission yeast. It's simple, it's only single cell, it only has 5,000 genes. We have 25,000 genes, but it means that it's simpler to understand. Now, how do you go about studying a process that you don't know, have any idea how it works? How can you do that in biology? Well, the way we can do it, and this is a fission yeast cell I've drawn here, is to use a chemical, for example, a mutagen that will damage the DNA. What we have here, these little green dots, represent a sample of the 5,000 genes in this yeast. If you irradiate or treat with a chemical, these genes become damaged. If any of these genes are important for cell reproduction, then cell reproduction becomes defective. In this case, instead of reproducing and dividing, they continue to grow, but they can never divide. And so they become highly enlarged. When I started this work in the 1970s, I identified 30 such genes. Now, because we've sequenced the whole fission yeast genome, we've deleted every one of those 5,000 genes, we now know there's 300 of these cell division cycle genes. But when I did this work, only 30 were known. Now, this shows you what those look like. Here we have a normal cell, that's the nucleus with the DNA. It grows, it divides, it goes around its cycle. Cell mass, cell size increases with time. If, on the other hand, it cannot reproduce, it produces these highly elongated cells, as you see here. So these are examples of, published in 1976 with one of my colleagues here, who's now the professor of biochemistry in Oxford, by the way. He was a graduate student working with me. These are what normal cells look like. These are these cells that can't divide. Do you get that? All straightforward. Now, these genes define processes that are needed for reproduction. But do you remember I said I wanted to know what controls reproduction, not simply the processes? Now, to explain that, we go back to this slide here, because what we see is an example of an important control. Think, you know, Italians love cars. Or so it seemed trying to drive here from Napoli, anyway. There were lots of cars on the road. Now, there are many components in that car that are needed to make it go, just like there's many genes that are needed to reproduce a cell. But you control the speed of the car with just one control, the accelerator. And so we were looking for the accelerator of the cell reproduction. Now, if you accelerate cells more rapidly through reproduction, then what happens is they divide before they grow enough. So these produce cells which are much smaller than normal. It's very simple. Everybody understands it, but nobody has done it before. So we looked in the lab for these mutants. I called them wee mutants because I isolated them in Scotland. And some of you may know that the word wee is the Scottish word for small. So these are called wee mutants. And this is what the first wee mutant looked like. See this here? These are dividing at half the size. You could not imagine this is important, but it is. Because what this does is define genes that are important for determining the rate at which cells go on to divide. So, I identified genes which behave like this, and these were important for controlling two major steps in the cell cycle. One Whoops, sorry. Oh, there. One controlled, a control set that controlled the rate at which cells underwent DNA replication, and another, the rate at which cells underwent mitosis, the two major events. But the extraordinary thing was, there was only one gene that controlled both those steps, just one gene. That was a gene I called CDC2, and it was a gene which ended up controlling the cell cycle. What did it do? It did this. It was a protein kinase, an enzyme that was a protein kinase. Protein kinases are enzymes that put a molecule called phosphate onto other proteins. When the phosphate goes onto the other proteins, the proteins change their shape. When they change their shape, they change their properties, and they can be switched on or switched off. So this type of molecule is a fantastic switch for switching events on. And what this CDC2 protein was doing, we call it cyclin-dependent kinase, I'll come back to that in a moment, is switches on S phase and it switches on mitosis. I've illustrated how that works here. You add a phosphate, there's the red blob, onto different proteins. It changes the structure of the protein, and it activates this S phase and activates this mitosis. CDKs, or cyclin-dependent kinases, have two components. One is this protein kinase, another is cyclin. I mention that because my friend Tim Hunt received this prize 9 years ago, and he discovered cyclin, the other element of this. So I'm linked always with Tim Hunt all my life. And he told me I must come here, by the way, if ever I get offered this prize. So that's why I'm here. He had a fantastic time here and particularly loved the food. Now, I don't want to say much more, but what I want to tell you is this is the cell cycle, and I'm going to tell you how these cyclin-dependent kinases control the events, and it is very, very simple. What happens is, it starts, if you think of the level of redness as the amount of activity associated with this protein, it starts very low, it goes to this level, and it promotes S phase. It goes to another level, and it stops S phase, and it goes to a very high level, and it promotes mitosis, and then the activity is destroyed, and it starts again in the next cell cycle. In other words, it simply is climbing up through the process of cell reproduction, undergoing these different events, destroyed, and goes through it again. It's the simplest possible model. I want to emphasize that simplicity because in science we always look for simple explanations. Often we don't find them in biology, but this is an example. Now, my last message. I've been talking about yeast, and who is really interested in yeast? Well, as it happens, I'm interested in yeast, but I do appreciate the majority of the world, and probably including most of my colleagues, are less interested in yeast. So I decided that we needed to see if the same control also worked in humans. So how could we do that? Well, what we could do is this, and this was the first time this had been done. I could take a yeast that was defective in this gene and therefore couldn't reproduce. Then I took a gene library from humans, actually the very first gene library that was ever made, not made by me, but I got it within a month or two. And the gene library is to take the human genome, cut up into little pieces, and essentially sprinkle the little pieces on these yeast cells. If humans had a gene that did the same job as the yeast gene, and if a yeast cell took up that gene, then the yeast cell that couldn't reproduce could now reproduce as a consequence of the human gene providing that particular process. This experiment really shouldn't have worked, because remember, yeast and humans diverged apart 1.5 billion years ago. The fish, the very famous fish here, is 65 million years old, but we are talking about a process that's been the same for 10 times longer than that. But the experiment did work. Yeast cells grew, and they grew because they were now being controlled by the human genes. That meant I could extract it back again, sequence it, and show that it was the same gene in humans that controlled human cell reproduction as yeast's reproduction, and that means that it's the same in every living thing that you can see, except bacteria, it must be said. So every other living thing is controlled by this process, and this is what led to the Nobel Prize. So, just to conclude, cycling-dependent kinases control the cell cycle. Increasing levels of it control the two main events of the cell cycle, S phase and mitosis. And it's the same in all eukaryotes, and it's been the same for 1.5 billion years. That's 1,500 million years. And it means that you can use simple organisms like yeast or flies or worms to study basic processes in humans. Thank you very much for listening to me.